Here we'll talk briefly about carrier proteins and how they aid the transport of hydrophilic solutes across the membrane. Before we do that, though, let's remind ourselves that lipophilic solutes, especially when they're small, can pass across the lipid bilayer passively. They don't require a carrier protein. Things like oxygen or carbon dioxide can readily pass across that lipid bilayer. They're small enough that they aren't repelled by the phosphate heads, and then they're soluble in the hydrophobic core, and then again they pass between those phosphate heads of the phospholipids to pass right through that membrane. And so they can just be driven by their gradients across the lipid bilayer. Now, when we start talking about polar substances cannot dissolve within the lipid bilayer. Things like electrolytes, like sodium, cannot just pass freely across that lipid bilayer. And so they require a carrier protein or, in some cases, a channel. And so a channel and a carrier protein are somewhat different. A channel does not directly interact with the solutes that are passing through it. So for instance, we just essentially create a pore, and then that ion can move down its electrochemical gradient across that membrane. Now, a carrier protein is different from a channel in that it directly interacts with, binds with the substance that it's transporting. And so we've got our generic carrier protein shown here in purple, and it's moving this round solute, and that can be energized by a gradient, once again, in which case we're facilitating that diffusion, right? We're aiding the diffusion because it can't occur passively across that lipid bilayer. It can be driven by ATP. When the gradient doesn't favor the movement of a solute that we want to move, then we have to put energy into that, right? And of course, that can come in the form of ATP, like the sodium-potassium pump is driving both sodium and potassium against their gradients, energetically unfavorable, so we have to put energy into it. Now, we could also use energy derived from the gradient of another solute. And in this case, we have to be moving two things at once. And that's either co or counter transport. In other words, co meaning the two things are moving in the same direction, or counter transport meaning they're moving in opposite directions. And that really just depends upon the static gradient of the substance that's providing the energy. Now, and whether or not we want to move that substance into or out of the cell. Now, what happens in this scenario is that one solute is going to move down its gradient and therefore be releasing energy. And that energy is harnessed to move the other solute against its gradient. Right? So again, we have to have energy when we're going to move things against their gradients. And sometimes that's provided by one solute moving down a gradient to drive another up. Now in our model here that we're showing, we have our solute that's interacting with our protein, the purple guy here, and we get this sort of like conformational change. In other words, in the process of binding to the solute, the protein changes shape and then expels that solute on the other side. And what we're showing here would be called uniport. And in that case, it could be driven by ATP. We're not showing that here, but it could also be driven by the gradient itself, in which case it would be facilitated diffusion.